Hi everybody, welcome to my YouTube channel. Today we will discuss about very simple topic like Maillard reaction. Maillard reaction is one of the food related reactions, means it will be deals with the proteins, peptides and amino acids. So we will discuss now slide by slide what is Maillard reaction, what is the mechanism of Maillard reactions, and some few examples and important applications of Maillard reactions we will discuss today. Now you can see here, before going to discuss about the Maillard reaction, we need to know what is protein. Proteins are the most abundant and fractionally adverse molecules in living systems. Every life process depends on the class of molecules like proteins. So protein forms the fundamental base of structure and functions of the life. How it possible? So, and you can see here, I was one of the image we recorded SDS page, sodium dosyl sulfate polyacrylamide gel, and this is the marker and these are the molecular weight. See, you can see here 1, 2, 3, 4 up to 10. These are the various different kinds of sample and M is the marker and this is a molecular weight. So you can see here 6.5 to 205. See, means the small molecules like weight, the SDS page is mostly depending on the molecular weight. So small molecular weights, molecule will move faster in down and bigger molecules will move very slow. So from this principle we will understood the how the our sample looks like and what is the mass of the our sample we will discuss from, we will know from the SDS page. Now we can see here what is Maillard reactions. Maillard reactions are reactions has been named after the French physicist and chemist Louis Camille Maillard in 1876 to 1936, who intentionally described it, and is often defined as the non-enzymatic browning reaction. So one of the important point is what is Maillard reaction. Maillard reaction is one of the non-enzymatic browning reactions. And while food are processed or cooked at high temperature, the chemical reactions occurs between the amino acids and reducing sugars, which generate the different flavors and brown color. This is the actual concept of the Maillard reactions. Now you can see when the food is processed or cooked at high temperature, the chemical reactions occurs between the amino acids and reducing sugar. And then you generate a different kind of flavor and other different kind of color, that is called as a Maillard reactions. So it is often used in food industry for going food different taste, color and aroma. Maillard reactions are initiated by a condensation of amino groups on proteins, peptides, amino acids with carbonyl group on reducing sugar, resulting in shift phase formation and rearrangement of amatory and Haney's product. You can see the Maillard reaction products are generated during the cooking and baking, often when the reducing sugar react with amino acids, peptides and proteins. They are found in high heat processed foods such as coffee, brew and bread crust and roasted meats. These are the few example of high heat processed food of Maillard reaction. So you can see the exactly what is Maillard reaction. So Maillard reaction is a chemical reaction that occurs between the amino acids and reducing sugar. The thermal process of amino acids and sugar produce the mixture of complex compound, though the reaction is responsible for the non-enzymatic reaction. So you can see this is one of the non-enzymatic browning and food like say cookies and biscuits, breads and sticks. And browning occurs during the frying and roasting and shearing and baking and different overall quality of food. So the product of the changes is sensory attributes such as color, flavors, aroma, side browning and alter the nutritional properties and global recognition. So these are the few important points about the Maillard reaction. Now you can see acrylamide is the byproduct of the Maillard reaction. You can see acrylamide one of the byproduct of the Maillard reaction. Means when the food is heat the process, the chemical reaction will goes and change their colors and aroma. So sometimes it will form a acrylamide. So acrylamide is one of the byproduct of Maillard reaction. It formed between the asparagine and the alpha amino acids found in food, and dicarbonyl acrylamide is found maybe food according to the International Agency of Food Research in Cancer considerable carcinogenic. So acrylamide is one of the carcinogenic, maybe it cause cancer also. So this is the few of the limitation of the acrylamide. So now you can see the what is factor affecting of Maillard reactions. So Maillard reactions affect the several factors, so which can be significant. Now you can see one is the temperature, and second is the time, and third one is the and pH, and fourth one is the water activity, reactant source, concentration, and type of ratio of reducing sugar and the food composition. These are the few factors affecting of the Maillard reactions. And you can see now what is the mechanism of Maillard reaction. Now you can see here what you earlier discussed here. You can see this is the glucose. So when you take our food, glucose interact with the amino acid like NH2 and combine to glycolamine plus byproduct, and byproduct is the water. So this is actual the first steps of the Maillard reaction. Now you can see the second step when the glucolamine undergoes the andromery rearrangement and form ketones amine. This is the one of the compound of the ketone amines. This is the ketone amines like and amidori compound. So these amidori compounds again goes the fusion product. So means it will be breakage and it will be reducer, and finally hydroxymethyl furfural it will be formed. So these are the important. Now you can see that you see the previous mechanism how is sugar convert into the glucolamine. Glucolamine again convert into that byproduct of the fural. These kind of reactions will be seen in Maillard reactions mechanism. Now you can see the importance of Maillard reaction. Maillard reactions can produce flavor aroma during the cooking process. It is used everywhere from the baking to the dairy industries, and it has important to our leaves of the reactions makes food tasty, and the roasted coffee flavor and yellow gold color and French fries and golden brown color of breads and the human in tasting this color of that Maillard reactions and conversion of caramelization one also cause that browning in food. However, this is the two different process of non-enzymatic browning. While the Maillard reactions involves in the amino acids, and caramel reaction are the caramelized product caramel candidates. They prepare the cream glue and the making the caramel, onion, carrots and apples. So these are the two methods, and you can see actually what is the mechanism of the Maillard reaction. Sugar, CHO and amino acid at the high temperature to form, it will be form a shift phase, and this shift phase you can see here. Then after antimony product adduct form, this is the colorless of the step 1. Then after it undergoes the dehydration and fragmentation, the product will form HM, F and the pyroaldehyde, and this is the step 2. And then after aldol condensation, melatonin is the step 3, and then form a different aroma and color. So this is the, you can see from top to bottom, you can see the process. This is the actual mechanism of Maillard reactions. Okay, then you can see here now what you discussed the previous proteins. The proteins are mainly here 3 classes based on the chemical composition: simple protein, conjugated protein, derived proteins. So simple protein is plasma, albumin, histidine, and conjugated protein is made up of like proteins and non-protein parts like hemoglobin, myoglobin and the egg albumin, casein. And now you can see the derived proteins, especially in derived protein you can see simple and conjugated protein, gelatin, collagen. Gelatin is nothing but of collagen and the byproduct of poultry industries, and the peptones and the protease. These are the derived proteins. Now you can see here the color reactions of proteins. You can see the like Bayreuth reactions in is one of the best test for the confirmation of a detection of peptide bonds. So either your your solution is either present or not of the peptide bonds, you can find out by root test. Then after you can see how the different linear enzyme and polypeptide hormones are metabolites of body, and collagens are from different calcium phosphate and contracting protein muscle fermented, and these bloodstream proteins such as the hemoglobin and plasma albumin are the subtle molecules essential for life. Now we can see the classification of protein chemical structure, functions and nutritional importance, and you can see actually the classification of protein chemical nature and solubility: simple, conjugated and derived protein, globular and pseudoprotein and glycoprotein, primary and secondary proteins. You can see different form. And the what are the exactly the what is the function? Based on the functions, proteins are the different types: structural protein, enzyme catalytic proteins, structural protein, collagen, keratins, and transport proteins, happy lipoproteins, hormonal proteins, contractile proteins, storage protein like myoglobin, and one of the genetic proteins and diffuser protein, the receptor protein. Now you can see the importance of these and colloidal properties. You can see here the hydration cells, electrical fusion, the makes proteins stable in solution. So electric repulsion. So you can see diameter of 1 to 100 nanometer in the range of the colloid. So the hydrophilic groups of the surface form the hydration shell, and electric repulsion makes the protein stable in solution. Now you can see here some hydration of proteins. Proteins when come into the contact with the water and absorb the water in a cell of, and then polar groups of surface form a hydration shell. The hydration shell electric fusion makes the protein unstable in solution. Electrical repulsion. Then after you can see actually this is the figure. You will understood completely what is Maillard reaction. Maillard reaction mainly you can improve these antioxidant activity, bacteriostatic activities, and bacteriostatic activity, and anti-tumor effect, and they can see anti-allergic effect and anti-hypersensitivity activity. We can find out by the Maillard reactions, and changes the color and flavor and nutrition. And you can see the control of the, you can control sugar, water and temperature, time, pH and sulfite and amino acid compounds. You can be controlled from these Maillard reactions. Antioxidant activity and bacterial stasis activities and bacteriostatic activity, anti-tumor activity, anti-hypersensitivity activity, anti-allergic effect. Then you can see that how these DNA, ribose and glucose, sugar, Maillard reaction at heat temperature, new flavor and brown color. The protein amino acids combined to form amino acids. You can see the brown. So DNA and ribose and glucose also will be like sugar form. This is Maillard reaction, high temperature change this. Now you can see actually this is the figure. We understood the proteins, and you can go the carbohydrates and Mandori reactions product, then form the Maillard reactions of product, color change. Then you can see here the how the protein and sugar combine will by and heat, then change the color, flavor and aroma of the baked breads. This is the actual mechanism of Maillard reaction. You can see flavors of product producing reactions like furan, furane, pyrogen and thiophene. These are the different meat and burnt and sweet caramel, cooked and meat roasted. These kind of flavor producing reactions in Maillard reaction. You can see here furan and thiophene in meat burns. Puran. Then you can see golden brown and delicates. You can amino acid overheat and they form the golden brown delicious. You can see the protein and sugar, heat, color, aroma. This is a mechanism. If you like this video, please like, share and comment. Thanks for your watching, for your spending time, and I hope you will be understood something. I am little bit of hurry. I was makes very short, and time was very consuming. So we will next class we will discuss a very detail about the Maillard reactions and different example of Maillard reaction, and especially in food chemistry how it is Maillard reaction applicable, and caramel reactions, and what are the DNA and protein structures how will change while the temperature will goes in heat. and please like, share, comment, and my YouTube channel. Thanks for watching. Thank you very much for your kind attention. We will see you next YouTube class with the detail about of this lecture or this modulation. So we will see. Bye. See you. Take care. All the best for your future.